Hi, folks. Welcome to the Grit City Think and Drink. Just so you know, this is being recorded. So if you have any friends that couldn't make it tonight and they're interested in Robin's talk, we will record this and make it available after the fact on the Think and Drink YouTube channel, which you can get access to from the same website that sent you to the wrong place just moments ago. So hopefully it'll work better when you go to see this recording later. Just wanted to let you know, so my name is Jim Gall. I'm the host of the Grit City Think and Drink. I'm a professor at the University of Washington in Tacoma, and I'm happy to have you all here tonight virtually on election night to talk about something that isn't elections, to get a little break. So thanks to Robin. I will introduce her in just a second after I talk a little bit about how this goes. For those of you who are first timers, we do this every second Tuesday of the month. And we hope that you will join us again in December. So December 13th is our next one. And Rachel Frick, who's a grad student at UW Seattle, is going to talk about how to use smartphone data to actually learn more about natural resource management. So should be really interesting talk from her. So we hope you'll join us on December 13th, same time, 6:30. It's a different link each time, and hopefully we'll get it right this time. So hopefully you'll join us again. And we have 2023 started. If you have ideas for speaker in 2023, we still have some slots open the second half of the year. So if you got people that are interested, please let me know. Send me their name or contact info, and I can get a hold of them and ask them to come be a part of this. So the way this is going to work is that I'll introduce the speaker in just a second. She'll give us the most amazing talk you ever imagined. Then we'll give her a short break. No pressure, Robin. We'll give her a short break and we'll give away some prizes. We have some amazing, if you can see these with my thing on, let's see if I get it near my face, Grit City Think and Drink socks, which are hard to see without me, and some other prizes. So we'll give those away in a trivia. And then after that, we will ask questions of our speaker, and you can ask whatever you like. I will moderate that, Robin, so you get a chance to talk about that, and I will feed you the questions so you don't have to pay attention. Just so you know, because of Zoom bombing potential, which we've had happen once, I hate to tell you, but your videos and your images are not being shown at the moment. Maybe later on we will turn it on if things feel safe and we know everybody in the room. But otherwise, sit back, enjoy, and I'm going to stop sharing my screen. And I'm going to introduce our speaker. And Robin, you can go ahead whenever you want to share yours. So Robin Kodner is an associate professor of environmental science at Western Washington University. She has been studying algae and biodiversity for 25 years. Her passion for playing in both the mountains and ocean has driven Robin to focus her algae research on field-based studies of how these populations, in their natural environment. Currently, her lab works on the snow microbiome from Pacific Northwest as a model to study how these communities evolve in response to environmental change, as well as studying microbial communities in Bellingham Bay and the response to changing ocean conditions over seasons and years. Hopefully a future talk. She founded and directs the Living Snow Project to enable collection of large-scale pink snow microbiome data and offer an opportunity to engage local communities in science through recreation. So thank you very much for being here, Dr. Kodner, and it's all yours.
Thanks so much, Jim, and thanks everybody for joining us on Zoom on this very exciting day. Hopefully it is a bit of a break from whatever else is happening out there. I happen to be in LA right now at a conference, and I have not looked at the news yet all day, so I'm happily ignorant about what is going on out there. Okay, so I'm going to talk to you today about pink snow and the Living Snow Project and what we are doing in my group and with the program to study how snow microbiomes are changing over time. And this is a more difficult thing to study than you might imagine. And so leveraging the power of community sourced science is really helping us tackle this large problem. So this shows the homepage of our website and the address to our website. And the Living Snow Project's goal is to engage the outdoor recreation community in research that is characterizing biodiversity of pink snow and the impacts on snow melt dynamics. And I'll tell you a bit more about that in a minute. But our philosophy is that by empowering people of all ages to participate in science in the places they love to play, we cultivate awareness of the living planet and a greater appreciation for conservation of threatens ecosystems. So we have this goal of helping people connect with the environments that they're playing in and understand that more from a place of understanding the science, as well as hopefully inspiring action towards conservation, but it also helps us do better science. We run the project out of Western Washington University, out of my lab, and we also collaborate with a group at the Desert Research Institute and have in the last 2 years done a focused effort of recruiting folks in the Sierras to have some comparisons between the Pacific Northwest and the Sierra Nevada ranges. So you can sign up to volunteer on our website, and you can, you can contact us there. And so what are snow algae? And sometimes people call these things watermelon snow. And these are a group of closely related microbes that bloom in snowpacks, which is an extreme environment. These organisms can withstand freeze-thaw cycles. And when they bloom, so when they're growing at high concentrations, the pigment that's in their cells, which is red, is visible on the snow surface. And that can have impacts on the rate of snow melt. So when you darken the snow surface, you decrease the rate of the amount of solar radiation that's reflected off the snow surface. And so, which is called albedo. And so just like any dark surface will decrease the albedo, when snow algae grow on the surface, they can decrease this albedo. And so this means that growing algae that are darkening the snow surface and blooming help increase the rates of snow melt. So that can be an added stressor to our snowpacks and glaciers in our region. And this article from the New Yorker from a few years ago said why the last snow on Earth may be red. And so this is a bit of a exaggeration perhaps, but maybe not. And so as we start to lose our snowpacks, we can imagine that the algae, which like wet snow, are going to grow and bloom in more places and at higher concentrations. And so when we look at algae in snowpacks, they can present on the snow in all these different ways. So you can get really intense blooms that can be spread out across a surface. You can get these little pockets of red. Sometimes through a freeze-thaw cycle, you get layering effects in the snowpack. So you can see that they can be found in all of these different forms on the snow. And when we collect them, they can be super high concentrations of cells. I'll give you a little bit of an orientation of what the Living Snow Project is. And the reason that I had the idea to start this project is because microbial populations are very diverse. And when you look at these pink snow patches, you can't tell which species are blooming just by looking at the snow. And so there's many different species that turn the snow pink, sometimes orange, sometimes red, and you can't tell by looking. And so, and they change over time. And so just because you've been to one place and you've collected, that doesn't necessarily tell you what's going to be in the same sample if you come back 3 weeks later, or what's going to be in a sample that you collect, 10 meters down the snowpack from where you are. So the idea was that by recruiting people who could collect samples across space and time to create a really robust data set, we would be able to better characterize who was causing these blooms and how they may be changing over time. And so the Living Snow Project allows people to volunteer in a few ways. So they can volunteer to make observations of pink snow, and they can also volunteer to collect samples of pink snow. And so to make observations, we have an app, and our app developer is here, who's done an amazing job at creating this resource. So you can just put the app on your phone, and it works on Apple and Android phones, and you are out on a trip in the mountains and you see pink snow, you can let us know that you've seen it. You can take a picture of it and upload it to the app. If you happen to have a sample kit with you at the same time, you can take a sample of this and upload the tube ID with your observations, and then people ultimately send their samples back to us, and we sequence the DNA in the tube. And what that allows us to do is it allows us to characterize the microbial community. And the tubes come with a DNA preservative so that the sample, once it's collected, is stable at room temperature for almost up to 2 weeks before we get it back into the lab and we get it cold. Here's a little video of me collecting some snow algae.
the way and you find the brightest pink parts. We've done 2. And then to be able to fit more in, and shake it so the snow mixes with the preservative, and then there's a bit more space and you just put more in. Look how bright this is underneath.
There we go. So that gives you a little idea about how we collect the samples. It's really easy to do. That was collected sort of a late season sample. You can see I was wearing running shoes. So I went on a hike and saw some snow and collected it. So we do have this, as I mentioned, this goal of engaging people in the process of science. It also empowers people to decide what gets sampled and raises. So that's an important piece of this, because somebody's going out to a place, they see pink snow. It's really a big bloom, for example, they're interested in that place. They can sample that particular site that they visited and then send it to us. So ultimately that gives them more of a connection to sort of the collective data set. And then that raises more awareness for just the process of science and what we're doing. We have a number of different scientific goals. So we want to study the biodiversity and biogeography of snow algae over time. And that's because these populations are diverse. And as I said, they're highly variable. And so if we want to be able to understand how they're changing with respect to climate change, we need some level of baseline data to understand just what represents the community, what are the species distributions, and then what is the spatial and temporal variation. So we need to be able to define all of these things before we can assess whether or not they're changing due to environmental change. So alpine systems where we work are dynamic. So they're both seasonally and annually dynamic. And many of you probably know this because we don't have the same amount of snow each year, and the snowpack doesn't melt the same way each season. And so every year is different, therefore the blooms are different. Not all colored snow is the same species, as I mentioned. Snow algae have complex life cycles, which you may not know. All algae have sex, which is an exciting piece of their life cycle, and we try to track down when that happens. That also allows them to have more biological diversity than other microbes. And there's strong connections between snow algae and their impact on snowmelt. So we want to understand if this distribution of snow algae across mountains actually is leading to changes in snowpack or changes in glacier melt. And if we can ultimately, when we can sort of define this system a little bit better, we'll be able to make predictions about future scenarios. So if we think about this dynamic system, we think about the snow algae growing season. So when people volunteer, they can start volunteering in the spring and start hunting for snow algae as soon as the snowpack is starting to melt. And then that early season, we have a lot of wet snow. By mid-late season, we start to get a very consolidated snowpack. And by the very end of the season, we start to have dry residual snowpacks that can still harbor snow algae communities. And so you can see this little diagram of what we think about in terms of how the growing season changes over time. So that's over a season, but then we see this process change annually. So this data shows you snow depths from the Mount Baker ski area. So this is near one of the sites that my group samples regularly. And this is 3 years of data where we looked at the length of the growing season. And you can see that there was quite a difference in the amount of snowpack in each of these years and when it started to melt, but the rate of snow melt was consistent. So once it started melting, it sort of melted at the same rate in each of these 3 years. And there was different growing season, the start and end of the growing season. And so there's a lot of variability that we need to take into account. And so this is just one site where my group goes and samples regularly, but of course we can't do this for every site. And so by sending a lot of folks out into the mountains by order of, just by the sheer number of samples that we get from volunteers, we're able to look at sort of when we find snow algae starting in the beginning of the growing season and then moving towards the end of the growing season with this power of community observation. And as I said, not all snow algae are the same. And so we know that certain sites are dominated by different species. So these are 3 different species of snow algae. This one dominates in the Bagley Lakes area. This one dominates in the Black Peak region. And then this species Sanguina dominates in Watson, the Watson area. So I wanted to tell you a little bit about their life cycles because this is directly connected to the growing season. So we think that the snow algae can repopulate the snow each year by swimming up the snowpack. And the way they do this is they have a part of their life cycle where they can swim. So they're a flagellated cell. Once they get to the top of the snowpack, they turn into a non-modal vegetative cell and they get their red pigment, which helps protect them from UV stress as well as oxidative stress, which happens when you do a lot of photosynthesis. Because when you do a lot of photosynthesis, you produce a lot of oxygen, which creates free radicals that wreak havoc on your cells. And so by having this pigment, that can help quench it. Just like when someone says, eat like beta carotene or something like that for an antioxidant to improve health. It's a similar type of principle. And so what we see over time is that in the summertime, we see the pink cells on the surface of the snow. And then through the fall, and then those cells become cysts at the end of the season. Then the snowpack falls on top of the cells over the fall and winter. And so they overwinter underneath the snowpack. And then in the spring and summer, when the snowpack is saturated with water, they can develop into their swimming cells. They can swim up the snowpack. They can divide, and then they can produce their pink pigment. And that is the bloom that we see on the snow surface. We do also know that algae can be spread by wind dispersal. And so when a lot of microbes can be spread through this process, and you might have a hard time believing this, but actually these cells can be blown up into the high atmosphere and transported over long distances. And they can also be transported over smaller distances within a mountain basin. And so the data that we collect with Living Snow Project is also helping us determine which mode of dispersal, the swimming up mode versus the aerially dispersed mode, is the dominant mechanism in different places. And I can say that in some of the places we see some real consistency in the communities year after year, and in other places we see a high amount of variability. And so we think that both of these processes are operating in our region. And so then how do we get the data? So people send tubes of collected snow back to us in the lab. We look at it under the microscope, and all of these are students that work in my lab at Western Washington University, and we extract the DNA from these samples. And then we make a DNA fingerprint for each sample. And that's what we use to characterize the microbiome. So we have a sample that's a mixed population sample. And so we extract all the DNA in that sample. And then each of the species has a unique DNA sequence for the gene that we're looking for. And so then when we look at all of the data that's collected, we can then match that up with the known DNA sequences from each of the species. And then we can get an assessment of the biodiversity in the species in each of the samples. So I want to show you a little bit of data because this is what ultimately we do with Living Snow Project data and what we can do with this sort of large amount of sequence data. So each one of these samples that we collect produces, when we sequence it, we get anywhere between 10,000 and 50,000 DNA sequences per sample. And you might wonder, how do we compare 50,000 sequences across 100 samples? It becomes a complex data problem. And what we do is we do a clustering analysis where each one of these points on the plot represents a single sample. And so points that are close to each other on the plot are very similar, and points that are far apart are very different in terms of their community structure. And so we did this analysis. You can see that there's a bunch of different shapes. Each of these shapes is a mathematical cluster. So we have sort of 5 different defined communities in the North Cascades from this first analysis of about 100 samples collected through the program. And each one of these clusters is represented by a different community of algae. And so the X cluster, for example, is this pink. Each of these pink colors represents a different species of algae. And so These are our 3 dominant species of snow algae in our region. There's a Chloromonas, Clanomonas, and Sanguina. And these are also can be related to where and when we find these samples. And so on the x-axis here is when we've collected the samples. So May is those early season samples, August are late season samples, and then elevation is on the y-axis. So what we see is there's a pattern in these clusters. So these 2 species, the Chloromonas and the Clanomonas, are dominating our early season samples, and our Sanguina are dominating later season samples. So if late in the season, like in July and August, you're up in a high snow field and you see a bright red, more than likely it's this species of algae. So from the start of the program in 2013 through now, and the majority of this data has come since 2017. We have over, well, this says over 698 samples, but now we have samples logged from 2022, and we're up to 780 samples in our data set. And these are color coded by where we're finding them. If this map on the left is zoomed out, because you can see we have a volunteer up in Barrow, Alaska. We have some volunteers in British Columbia and Alberta, but the majority of our volunteers come from the Pacific Northwest region or this targeted recruitment in the Sierras. So I want to tell you a little bit about what this data looks like if we focus in on one particular place. And so we've been working on the Bagley Lake system. So this area is right next to the Mount Baker ski area. So it's essentially in our backyard at Western Washington University. And this is a snow on lake system. So this is snow that sits on top of a lake. And what's cool is that the algae bloom on this snow that's on top of the lake before they bloom anywhere else. So we have this very defined bloom we can study. And it's dominated by a single algal species, which makes it easy to study. And the annual snowpack on this particular habitat disappears completely every year. So this is early season on the left, and this is later season on the right. You can see the snow is melting, but then by middle of the season here, there'll still be surrounding snow in the area, but there'll be no snow left on the lake. And so when we go back to this place year after year, and again, Living Snow Project data helps us do this type of work where we're revisiting sites year after year. What we found was when we looked at the area that's next to the parking lot, we looked at the area that's right on top of the lake, and then the area that's next to the lake, that we have 3 different communities. And in fact, our lake samples are dominated by there basically is only one algae that's growing in those samples, where the samples in the parking lot and the samples in the adjacent hillside next to the lake are actually far more diverse. So each one of the colors on these boxes is a different algae species. It turns out also that this pink algae species is the Clanomonas species. It's a new species. So it's not a very exotic locale if you're from Washington. Thousands of people go up and backcountry ski in this region every season, and we've been skiing over an undescribed species of algae. So we've been going back to the Bagley area every year. This is from 2021, where we did a focus study where we went every single week. And so you can imagine if people are going out every single week and collecting samples, each one of these bars represents a replicate sample. So like on the first day, we collected 3 samples. On the second day, we collected 8 samples, and so on. And what, in this case, the pink taxa here is now red, but it still dominates, but it changes across the season. So when you see colors come in, like these brown colors or these yellow colors, these are different organisms that can possibly eat the algae. So this brown one is a ciliate that can eat the algae. This yellow one is a algae parasite, which appears early in the bloom and then kind of has a peak in the middle. And then at the end of the bloom, we see the rise of fungi that are taking advantage of the carbon that the algae is fixing that is then in the snowpack for them to eat. So we do see quite an interesting seasonal change in the microbiome over time. Okay, so how am I doing on time, Jim? I think I have about 5 more minutes maybe to go to our 30 minutes. So
you can still take 10 minutes if you need it.
Okay, Great. So I wanted to remind people how to participate, and I wanted to tell people a little bit more about what you do when you participate. So now that you know what we do with the data, hopefully that's an incentive to want to participate yourself. And so first thing to know as a volunteer is how to recognize a good spot to make an observation and collect a sample. And so the first thing that I do is I survey the scene, I look around, I see sort of where it's pink, where it's not, and then find where I'm going to sample. So here's a video of that Bagley Lake site where you can see that there's not really any pink where I'm standing, but there's pink down on the lake. It also helps to walk around and see where you can find the brightest pink because it's often not evenly distributed across the snow surface. These are just different ways. Here's a picture of the Sisters Glacier where you can barely tell that it's pink, but it is pink and it's light pink. And we still like those samples because we're interested in when it's really dark, but also we're interested in just the presence. And when you take pictures of the algae and upload them to the app, we can then have an idea about sort of what the algae look like when they were blooming and to compare that to the species that we find in the DNA. Here's just a few more examples. This is one trip that I did where we had collected samples on the northeast and the southwest side of Mount Good. And the snow algae were presenting really differently on these two sides of the mountain because the snowpack was different. And you can collect on glaciers, you can collect an annual snowpack. So this person was out skiing. So the snow really looks a lot different when you collect it. The pink snow looks different when you collect it in the early season than when you collect it in the late season. And again, here's just a montage of different ways that the snow algae can be found on the snow. And my favorite snow algae collecting partner here, who's probably collected more snow algae samples than most people. Okay, so we find, if you're wondering about, well, should I take more than one sample on any given day, it really depends on what you see. And so this was one trip where I saw three different kinds of blooms that were on different places as I traveled from one place to the next. And so oftentimes collecting a transector, starting in one place and collecting samples as the snow changes can be useful. So to get started, you first sign up, then you get the app, then you get your kit, and then you start hunting for pink snow. So this link is old, but it'll still work. And so people who are on the call that want to sign up that haven't in the past, this one is still active. We send a new link out to volunteers every year. So this is the very end of our season. So as soon as it started snowing in the mountains, which was about 3 weeks ago, that's the end of our season. And so we won't be ramping up to recruit new volunteers really until the spring, because you won't be able to start collecting until late March at the earliest, mostly April. But if you're interested, you can sign up there and we'll know and we'll keep you on our list for next year. You also get to tell us whether you want to make observations or collect samples or do both. When you collect samples, you use the app to make your observation at the same time. But it's not everybody has like has a sample kit with them when they're out, or not everybody wants to take a sample and carry an extra thing in their pack, even though the sample collection kits are quite light and easy to use. You can just make observations. And we offer places where people can pick up kits. And so in the Seattle area, you can pick them up at Patagonia downtown. But if you also live in a place where you're not close to, in Bellingham, you can pick them up at Backcountry Essentials. We work with Patagonia in Portland, and we've been working with the Rock Gym in Wenatchee, amongst other places. And we have a number of pickup places in the Sierras. But if you're not close to a place where you can pick up a kit, you can leave your address for us and we will mail you a kit. You can download the app at the Apple Store or the Google Store. And this is a picture of the interface with the app. And you can tell us when you're taking a sample or making an observation and upload your pictures.
Going to make an observation. So you're going to press the plus sign in the upper right-hand corner. And then the first field says I'm taking a sample. If you click it, you can say I'm reporting a sighting. Done. It automatically logs the coordinates. So all you need to do is upload if you want to, but you could also add photos, and you need to take the photos ahead of time. So I'm going to take a photo of this pink spot here. Go back to the app. I'm going to select a photo from my photo library, finish, and then the photo's in there. And then you just press the little cloud button in the upper right, and it will upload, and it'll say upload successful when it's uploaded.
And it'll store the observations until you're back in cell reception if you're taking sample or you're making an observation when you're not in cell reception. And our newest version of the app allows you to tell us what color the snow is when you collect it and how big the bloom patch is. It has you estimate it based on, is it the size of a shoebox or is it the size of a school bus kind of thing. And the images really matter. So getting something in your image to scale really helps. And so this is an example of me putting my eye sacks in the picture so that I can sort of estimate the size of the patch just from looking. You can also use other things like your ski boot, or in this handy case, we had a notebook with a ruler on it. Our sample kits look like this, and they're easy to pack up and mail to us. We ship them out. We ask volunteers to mail them back to us. But if anybody has challenges with the cost of shipping, we have them let us know, and we will send them shipping. Here's a set of images of different volunteers collecting snow at different times. We really want to pack the snow in there. So when people scoop it, they put on a glove first, which minimizes the contamination from your hand. And we found that all of our Living Snow Project samples look really good. We don't see any evidence of contamination, and we don't see any difference in the quality of samples that are collected by volunteers from those collected by the scientists that work in my lab. And then we already watched this video, so we don't want to watch it again. And we have this wonderful, an undergraduate in my lab did this wonderful illustration of the steps involved in making a sample or collecting a sample, and that is included with each sample kit. And then the last thing I'll mention is we really like it when our volunteers share their photos on social media, and they can use hashtags Living Snow Project or the watermelon snow icons so that we can see people doing it. And we would like to do, one of the things that I'd really like to do in the future is do a better job of building community around participating in the project with our volunteers. And so a lot of that will come with future development with our app, having map resources on the app and allowing people to share more about their observations. So that will all come with time. And with that, there's a lot of people to thank who have been involved in this project. I have some colleagues that help me with the science bits. We've had a number of student interns. I want to call out and volunteers. I want to call out Chris Chapin, who is here, who is our app developer and has done such an amazing job on that. And lots of other students who have been involved over time. And I look forward to answering people's questions about Pink Snow or the Living Snow Project.
Thank you very much, Robin. That was awesome. A virtual round of applause, however you can do it on your own Zoom app there. So we will get to the stump the speaker portion of our talk in just a moment when you get to ask Robin her questions, but we want to give away a couple prizes real quick. So the way we do this, if you haven't been to the Think and Drink before, is that yours truly comes up with a couple factoids that are only loosely based to our speaker's talk, because I don't want to try to be, one, I'm not an expert in what our speakers do, and it would just, and if they actually already came up with the answer, you probably saw it in her talk. So our questions today are going to talk a little bit about snow. So be ready. So the way that you answer is you chat your answer to in the chat function. So get ready with your chat. So the first person to get the right answer gets the prize. And so once we get the questions get in there, you can also ask questions, but for now we're going to give away a couple of quick prizes. So first one, in Washington State, We have one glacier that's actually actively growing at last time I checked. So what mountain is that glacier on? A Rainier. Dan gets it. Dan Carlson, Mount St. Helens. That is correct. It is actually the Crater Glacier in the new crater of Mount St. Helens. That is one of, as far as I know, the only actively growing glacier still in Washington State. In fact, I think it's the only actively growing glacier in the lower 48 as well. So congratulations. So to win your prize, by the way, Dan, you need to chat, since nobody else but Robin and I can actually see it. If you can send your mailing address, because otherwise I have no idea, no way to get this to you. So if you can send me your mailing address, I will send you your prize. Your prize, by the way, happens to be a Mount Rainier hiking book. So, or sorry, Mount St. Helens hiking book. So you can actually go check it out and potentially go get a sample for Robin and her crew while you're out there. I don't think you're allowed to go into the glacier in the middle without special permission, because things fall on you inside that crater, but you can get it on the snow fields outside. All right, second prize is going to be a pair of Think and Drink socks. So your question is, Washington State set a record for snowfall in one season on what mountain? What mountain was that? Oh, we already have it. Mike Knopp gets it from Mount Baker. That's true. Special prize. I can give away another pair of socks. Anybody know how much snow that was? It was in 1998, 99, right before I moved to Washington State. How much snow fell that year? If you get close. Peter, you have it. It is 1,140 inches of snow fell that year. Pretty spectacular. So Peter, Dan, and who was the other person? Mike, if you guys can chat me your addresses, I will get you your prizes. I apologize on the socks. We only have one size left. So if they don't fit you, hopefully you can give it away to somebody you love. So that's your choice. So thank you all for that. And before we get to our questions, just remember this, our next talk is the second Tuesday of December, and we hope you'll chime in to hear a little bit about how we use social media now to collect data on how to manage public land. So without further ado, we're gonna look for, I know that we're already some questions in there. So you ready for your questions, Robin?
Ready.
Here we go. So Andy asks, one of the slides suggested the pink pigment only occurs during part of the life cycle. What is the function of the pigment? And I think you mentioned that, but why is it temporal?
So when the algae are in this, well, some of the algae has it. Let me see, maybe I wasn't ready to answer the questions. I'm just kidding. I'm ready to answer questions. So the bright red pigment helps with oxidative stress, and it also helps with UV stress. So just in general, it's a stress response, helps them live in the really harsh environment that they live in. When the algae are swimming cells, oftentimes they're green. So and these are green algae. So even though they're red colored, they are in the green algae group. And so underneath the red pigment is chlorophyll. So it's kind of the opposite of what's happening with the leaves right now, where you see the leaves turning red, where they were once green, and the chlorophyll breaks down and you see the carotenoids underneath. This is the opposite of that. So they add that red pigment to protect themselves once they're in that life stage that lives on the snow. When they're in other stages, like the swimming stage, we believe that that's ephemeral, and that's when they're swimming up the snowpack and they don't need the pigment yet. It's energetically expensive. It's hard for the algae to make the pigment, so they don't do it until they need it.
Cool. Grace asks, is the red colored algae the only color of algae that occurs on the snow, or is that just the only time we see something?
Yeah, so we know that the algae are there even if you don't see, sometimes when you don't see it as bright pink. And so if we collect just adjacent to bright areas, you can see it kind of grayish color. There's often cells there. Some of the species actually turn the snow orange. So I could go back to one of my slides and show, let me do that because I think it would be nice to see that This particular species that looks kind of yellowish here, when it blooms, it turns snow kind of orange colored. And this one can also turn the snow kind of orange colored. And there's different pigments in these ones than in the red ones. In Washington state, we tend to have a lot of bright red blooms, which are either this species or this species.
Cool. Audrey
I
asks,
actually I have one other question in my chat that's related to that. And one of our volunteers, Chris, found a kind of rainbow colored. So he actually found like a transition between green and orange and red, all kind of in the same patch. And that's really interesting. We saw a lot more of that this year than I have seen in recent years. And I think it's because our snowpack was super wet this summer. And it was really dark. Do you remember June? It was terrible. It rained all the time and it was really dark. And that meant that we were doing field work in the rain. And so those algae weren't making the pigments quite as quickly. And so we saw sort of a cycle in the life. The life cycle was cycling in this maybe slightly different way due to the environmental conditions.
That's really cool. I can imagine how much fun it is to post hole through mushy snow while trying to collect your samples as well.
Yeah, no, it's wonderful.
Let's see. Audrey asks, does the algae relate to black carbon found on snow?
Oh, that's an excellent question. The black carbon also darkens the snow surfaces and is a problem for changing the albedo of snow and glaciers. There aren't a ton of studies, but the ones that have been done show that there's not a lot of interaction between the black carbon and the algae. The algae can be stimulated, their growth can be stimulated by adding carbon. So they do photosynthesis, but they can actually use carbon to grow. But the black carbon is what it's what we call recalcitrant. It basically is not reactive. And so these algae can't really be like fertilized by the black carbon as far as we know. That's a great question. Theoretically, if you get a bunch of black carbon sort of blanketing a bloom, that could potentially shade the algae, or if you get like a lot of dust on the snowpack, like we experience when we've been having big fire seasons. So yeah, those are like pretty open questions.
Cool. Leanne asked, can you say more about the significance of the new species you found?
Well, it's exciting because it means that there's a bunch of undiscovered biodiversity. And so that is fairly true for many environments that haven't been well studied. So we go to these environments where there haven't been a lot of studies, and we use our DNA techniques and we find new sequences. And so this species, we also can tell just by looking at the shape of the cell and the size of the cell that it's different from the ones collected in other places. So that's exciting. There are not very many species within this genus, only like really sort of 3 described species. And the most closely related ones to our one in Washington are a species in New Zealand and a species in Japan. And this is all sort of work that me and colleagues in those 2 places have kind of been putting together over this last year. And that's really exciting because it looks like there's this Pacific Rim relationship between these species. And then there's another species that lives in Europe, and it is clearly different from the Pacific Rim species. So there's more of a biogeographic signal than we might have anticipated. And it's really cool.
I was actually thinking about this because it happens with a bunch of other species. Is there a chance for people walking into the backcountry to be spreading or bringing outside organisms into a new area?
Yeah, I think about this all the time. And I gave a talk for NWAC a bunch of years ago where someone asked me if the snow algae cyst could get stuck on your skins, like your backcountry ski skins. And I think that that's probably also true. So whether people are backcountry skiing or hiking around, they could be moving cysts around with their feet. It's really difficult. Using the DNA tools we have is pretty coarse. It just allows us to see what sort of at a high level, which species, but to actually track, like if we've moved some from one place to another within a region. We need some finer scale tools to tell the difference between populations. It's kind of like how we tell the difference between COVID variants, like they're all COVID, but there's slightly different variants and we can tell by sequencing their genomes. Same thing, we can tell like the different variants that would be from different regions. So we're working on developing those markers right now so that we can tell the difference between populations, but that's just, it's a new project and we haven't fully developed the markers yet. So it's an interesting idea and we just can't measure it yet.
Cool. Yeah, that sounds really interesting. I'm really curious about that kind of idea of how you can track it, but I understand it's a totally different kind of DNA testing or RNA testing to try to look at variants from populations, but that would be really interesting in the future. Dave wants to know, how do the algae survive from year to year when all the snow's gone? Do they survive as cysts, and what happens if you don't get snow one year?
That's such a good question. That's also something we're really interested in. So in the Bagley Lake study site, we know that those algae make cysts, and those cysts can be found in the sediments at the bottom of the lake. And we're looking at through sediment cores to see how that might have changed over time. We do know that even if they're not on top of a lake, that the cysts can like hang out on rocks or vegetation that's underlying the snow. And we believe that it kind of just hangs out there and stays dormant through the summer season and the winter season, basically until the snow is habitable again. And again, that habitability window is really small. And we would love to know what happens if there isn't snow for a year. Like, do those cysts, can they just hang out as cysts for more than 1 year, more than one season? My gut feeling about that is yes, that they have, they're pretty resilient once they're in that sort of cyst or spore form. And that's also, I think, how when they stay as cyst uncovered for a long period of time, how they also can blow around in the wind. And we've had volunteers ask us this question. And so a few volunteers this year have actually gone back to a place where they sampled and then sampled dirt or rocks in the same area that they had collected and sent us those samples, which was awesome. So we have the one place that we sort of have our focus study, but that's something that we could sort of leverage volunteers for also in the future.
Very cool. Let's see. I'm not sure if you can hear me because I'm saying my link is unstable, but Zinnia wants to know, is this a new phenomenon, or is the algae now showing because of global warming and ice melt, or is it showing more because of that maybe?
Yeah, so it's not a new phenomenon and it's natural. So like snow algae have been on snow for a long time. Evolutionary biologists, we can sort of anticipate the age of a group by using the DNA to sort of look at evolutionary relationships. It's a pretty ancient group, so they have been around for a long time. Now, to answer the question about if the blooms are getting more intense now due to climate change and increases in snowmelt is an excellent question. And you need a lot of data to be able to answer that because it's so variable from season to season and from place to place. And so we have like a lot of data from about the last 6 years. My hope is that in a decade's worth of data, we won't be able to see sort of a shift in elevation. So we look at all the places that people are finding samples, and then does that sort of average elevation shift up over time? And that's how we would be able to have the data that would confirm that they are in fact getting worse over time. But that life cycle that I mentioned, it's really dependent on there being liquid water in the snowpack. And so the more liquid water in snow or on glacier surfaces, a good hypothesis is that you would see more algae blooming.
Cool. We're almost out of time, so I got one more question for you. Ashley asks, are there areas living snow has few samples from? Do you and your students attempt to collect those? Are the ones that you recruit volunteers for? And where are those areas that you want to get samples from?
Yeah. So one of the premises that I've used to run Living Snow Project is that I want people to I want it to be easy. And so I just say, go wherever you're going. Like you're going out on a trip, just take a sample kit. If you see it, you can collect. What I would hope, and so sometimes, so that's the first answer is like, I just want people to go and collect where they're already going. What happens is that we end up with repeat samples from a lot of places because there's popular areas to go. There's popular hikes to go on. For example, Glacier Peak is a place where there's a lot of snow algae blooms. Glacier Peak is also a little bit difficult to get to if anybody's been there before. It's about a 15-mile hike into the mountain itself. And, you know, I could go there maybe once a year, but I can't go there every week. But volunteers actually visit Glacier Peak all the time. And so we end up having a lot of high resolution of sampling at Glacier Peak, which is awesome. And that's totally enabled by having volunteers collect. So that's my second answer. And then the third part of that is that you can see where we have sampled on our website. So there's an interactive Google map, and any place that you don't see samples from, we love. So we love it if people are off in some really remote place and they want to collect a sample. And then that adds to our resolution. We tend to, again, have samples like around places where there's trails and certain hikes and climbs that people want to do. And then usually if there's things that I'm particularly interested in, we'll tell volunteers that at the beginning of the season. So I'm really interested in snow that's on top of lakes. So I want to know if this new species that we've characterized from Bagley Lake is on all of the snow on top of lakes in Washington. And so that's another sort of cue that I can give volunteers that if you happen to be in that kind of place. And that's really an early season phenomenon. So a lot of backcountry skiers would be collecting for those types of samples. But yeah. And if somebody likes to go to really, really remote places and wants a challenge, then you can just send me an email and I'll tell you all of my dream samples.
Well, I think from what I saw, like Mount Goody, you're getting plenty of distance away from anything close to a road. So I think you're doing some serious distance yourself. But thank you very much for an awesome talk, Robin. It was great to meet you tonight. For everybody here, a big thank you to our speaker tonight, and thank you all for showing up and finding your way here from the ghost Zoom room on the other side of the world. So I appreciate you guys making it. Again, hope to see you in December, and otherwise this, as somebody asked, this recording will be up on YouTube. So if you go to the Think and Drink website, you'll see a link to all our past talks that we've been recording since COVID started. Including Robin's talk, if you want to check it out and you missed part of this. So thank you very much, Robin, and have a great time in LA for the rest of your conference.
Yeah, thank you so much, Jim. Thanks for the invitation. And thanks to everybody for showing up tonight. This was really fun.
Take care. Bye.
Bye.